Hello, everyone. I am Kenny Coogan, the Education Director for the International Carnivorous Plant Society, and we are very excited to have Dr. Qianshi Lin talking about a new carnivorous plant group, Triantha. But first, I wanted to give you a little background on the International Carnivorous Plant Society. We have Facebook and Instagram. If you go to our Facebook page, you will see all of the webinars and virtual happy hours that we have scheduled now through November. If you're a member of ICPS, you can go to Club Express to get the webinar links. A couple of months ago in May, we had our World Carnivorous Plant Day. We have over 24 videos posted on YouTube and Facebook for your entertainment and education. Part of that is every year we have a big photo contest. You do not need to be a member to enter, but if you're one of the 3 winners, you get a 1-year membership to the International Carnivorous Plant Society. You can submit your photos from January through April. So right now you can start taking photos to get ready. World Carnivorous Plant Day is always the first Wednesday in May. If you would like to be a speaker or if you have an idea of who should be a presenter for next year, please let me know. Next year, it's going to be Wednesday, the 3rd of May, 2023. Part of the Society and World Carnivorous Plant Day is that we have merchandise. We have totes and t-shirts and mugs, and all of that money goes to the education and conservation grants. Part of the education grants are the Carnivores in the Classroom grant. We did this last year. We were able to fund 24 classrooms, and they were able to add carnivorous plants, terrariums, tanks, grow lights, bogs to their classes. The application window is going to open up in just another week or so. It'll be open all of August 1st. If you would like to help fund Carnivores in the Classroom, you can go to carnivoروusplants.org/donate. All right, so without further ado, we're going to have Dr. Qianshi Lin talk about this new genus of carnivorous plants.
Thank you for coming to my talk, and thank you for Kenny to introduce me. And I'm Dr. Qianshi Lin. I'm currently in Penn State University to do the research on the evolutionary developmental biology of carnivorous plants. Today, I would like to show you my research on the Triantha and new carnivorous lineages. And I will first introduce how we found this Triantha is a new carnivorous lineages with a unique sticky florence trap. And then I will talk about the phylogeography and species circumscription in Triantha. So as we know, there are about 800 species of carnivorous plants right now. So most of them are in eudicots and only 4 are in monocots, and all of them are in poalis, and use the enlarged leaf rosette to accumulate water and drown insects in. And there's no clear evidence to show any of them produce their own digestive enzyme, which indicate that they may just be proto-carnivorous plants. So the question would be, why would you even think about to find a new carnivorous plants in monocots? So the whole story went back to a former master student in my lab, Greg Ross, and he did a phylogenomics of Alis matalis. And he found that Triantha lost some of the plastid NDH genes, and these genes present in nearly all land plants except some heterotrophic plants and carnivorous plants. And he found that the theme of occidentalis are covered in glandular hairs, which makes him thought that it might be possibly a new carnivorous plants. And then he graduated and I started my PhD, and I thought it would be a very cool project to carry on. And then the first thing I did was to go to the wild to have a look at the plant by myself. And I went to Cypress Mountain in West Vancouver, which is actually very close to the city, and in the summer, and you can see that the green one, just like lily, is Triantha, and the red one is the round-leaved sundews, and it suggests that they are growing in the same habitat as other carnivorous plants, and which might be new carnivorous plants. And we also found lots of insects on their flower stems and even on the herbarium samples, specimens. But to prove that it's carnivorous plants, you have to prove that they are getting nutrients from their captured insects. And to do so, I use the stable nitrogen-15, which is the isotope to the much more abundant nitrogen-14 to label the fruit flies. And I put the fruit flies on the stem of the Triantha and the sundew, which we already know is carnivorous plants, and the oregano, which is the non-carnivorous controls. And after 1 week or 2 weeks, we measure the isotope level in their tissues. And here is where we did the experiment on Cypress Mountain in West Vancouver in a big bog. And you can see that from the results, we see that after 1 week or 2 weeks, there's no significant increase in irregular and negative control. And there's already a significant increase after 1 week in sundew and a more increase in 2 weeks. For Triantha, there's a little bit increase after 1 week, and after 2 weeks, it reached almost the same level as the sundew, which shows that they are indeed getting nutrients from their captured insects and prove that it's carnivorous plants. And we are also curious about what they use this nutrient for. And when you compare the isotope in leaf and fruit, you can see that the fruit is actually accumulating the isotope, nitrogen, which shows that perhaps they are using the nutrients from the animals to supplement their reproduction. And the next will be if they can produce their own digestive enzyme, which makes them holocarnivorous plants. If they cannot produce their own digestive enzyme and have to rely on other organisms to do the digest, they will be the protocarnivorous plants. So I use a phosphatase substrate to put the phosphatase substrate on their trichomes. And if there's any digestive enzyme, it will break down this substrate and produce fluorescence. And you can see that under the fluorescence scope, the trichome of sundew and the Triantha is shining with a green light, which means that they are indeed producing their own digestive enzyme and makes it the holocarnivorous plants. And this is also the first clear example of holocarnivorous plants in monopods. And we all know that most carnivorous plants want to put their flowers far away from their capture sites because they don't want to accidentally kill their pollinators. Since Triantha use their flower stem, which is very close to their flower, to be the capture sites, it might have this kind of dilemma between carnivory and the pollinator. And the dilemma between carnivorous and the pollinator. And according to our observation, we can see that most of the they are pollinated by the big insects like butterflies and bees, and their trichomes is not that sticky enough to capture those big insects, which may just avoid this conflict between carnivory and pollination. So for this part, we know that isotopic evidence shows Triantha is a new carnivorous lineage, and the enzyme test shows Triantha is holocarnivorous. And after that, I want to do a population level of study for the whole gene and to investigate their gene loss. And if we still remember the whole story study with placid and DHA gene loss and in Alice Matalis, this gene loss has been found in submerged seagrass. And another example is in Tophidiaceae. And the function of this plastic dish has been now mysterious, but it's becoming clearer now, and it's involved in cyclic electroflow around photosynthesis 1, dealing with the stress response. And in Tophidiaceae, both species, investigated species, Scrutinosa and Occidentalis, has this kind of genus, but there are also different number of genes. So if we do a population level of study, we can find that different population may have different level of genus, which may let us to track back the origin of this kind of genus. And also why I want to do is because there's a taxonomic problem in this group. And there are only 4 species in Triantha. Occidentalis in West Coast, Scrutinosa in North and East, North America, and Rasmussa in Southeastern America. And the last one, Japonica, is endemic to Japan. And for 3 species in North America, the southeastern species Rasmussa is very easy to distinguish from other 2 species. They have that kind of styles which merged to the base, and they have the hard capsules, whereas another 2 species have that kind of the very spongy capsules. The really problem is to distinguish Occidentalis from Glutinosa, and the only confident way to distinguish them is by the seed coat. And the Occidental have this kind of spongy and white seed coat, whereas the Glutinosa has that kind of tight and brownish seed coat. And with those seed coats, there are other characters to suggest, has been suggested to distinguish them, but most of them are overlap with each other and is not diagnostic. So on the West Coast, there are some populations which has this kind of tight and brownish seed coats, just like Glutinosa has been identified as Glutinosa, which makes the Glutinosa have some kind of weird disjunct distribution to the West Coast. And to make things even worse, there are 3 subspecies in Occidentalis. We have from Alaska to Oregon, the subspecies Brevistyla, which has shorter styles. And we have subspecies Occidentalis in California, which has a globose inflorescence. And we have the subspecies Montana in US Rocky Mountain, which has longer granular hairs and other 2 subspecies. But most of those characters overlap with each other, and it's kind of still hard to distinguish them, especially when they are overlapped with each other. So to do a population level study, I collect many samples by myself and also request specimens of Triantha all across North America from more than 30 herbaria. And I designed the NDH genes primers for all 11 NDH genes and did the PCR. Here's the results. And you can see that the species Rasmussa and Glutinosa are well separated to be monophyletic, and the Japanese species Japonica is well nested within Occidentalis. We suggest that it should be treated as Occidentalis, and if we do so, Occidentalis itself is monophyletic. And we indeed found many possible misidentification on the specimens. And for all species, all populations on the West Coast which has that kind of tight seed coat, just like Glutinosa, they're all grouped within the Occidentalis, which suggests that they should be treated as Occidentalis. And the seed coat seems to be not that diagnostic as suggested before. But if we have the biogeographic distribution of Triantha, they are still well separated. And it's safe to treat all Triantha on the West Coast as Occidentalis and all population east to Rocky Mountain as Glutinosa. And they overlapped on the Rocky Mountain region. But on those regions, there's no tight seed coat type of Occidentalis, so we can still use the seed coat to distinguish them from each other. And I think the most interesting results is the placement of the Japanese species Japonica It's well nested within the Occidentalis and is sister to the population on Haida Island. And the question would be how Triantha went to Japan from North America. And one possible explanation that they may went to Japan across the Bering Land Bridge, and the last glacial period just erased all similar population on the continent. And we knew that we have known that Haida Islands is a glacial refugee, so perhaps some similar population still survived on those islands. And at last, let's have a look at the gene loss pattern. So we indeed found that different populations have different level of gene loss. So Rosmosa don't have any gene loss, Cutinosa have 1 or 2, and Occidentalis from none to almost lost all their NDH gene loss. The reason why they are losing their NDH genes is still unclear, perhaps because NDH genes is dealing with stress, and carnivorous plants may use nutrients from their captured animals to deal with this kind of stress and makes NDH not that necessary. And we also suggest there might be a link between the origin of carnivory and the gene loss. Because Rasmosa has the least sticky stings in this group, and we can see that they are not losing any genes. And perhaps Rasmosa is not carnivorous. And for Glutinosa, for Occidentalis, which has the most sticky hairs in this group, they also have the most gene loss. So there might be a link between gene loss and the origin of the carnivorous, but which need to be investigated in the future. And for this part, we know that NDH phylogeny address species boundary and the NDH genes exerting various degree of loss or already frame interruption in these two species, and there a strong geographic signal is evident. And I need to thank to many of the herbarians who are willing to sharing their precious samples with us, and all the collaborators and the people who helped me to do the field work. Thank you very much.
All right, that was wonderful.
Yeah, thank you.
I appreciate it, and so do the participants. We're excited to post it on our YouTube page in a few days. We have just a couple of questions. Do all carnivorous plants have NDH gene loss?
I think some of them have, especially in some of the Sandu and the Pinguicula, we found some NDH gene loss.
And how do you think gene loss influenced carnivory?
I think it's more like another way, like the carnivory influence gene loss. But the reason why they're losing these genes is still unclear. We have to investigate what the genes is exactly doing. So their function is still kind of mysterious and not very clear for us.
Every other year we have a international conference, and this year in May or 2023 we're going to be going to Japan. So maybe we can see Triantha in the wild there. Yeah,
they have the Japonica, but I think we lump them into the Occidentalis right now. They should have some name change. Yeah, we are trying to publish this paper.
Okay. In terms of conservation, is the Western species and subspecies, are they in danger from the wildfires and climate change, or are they in these microclimates of wetlands that aren't really affected, or should we be concerned for them?
I think they're not in danger right now, just like other, I think it's not the plants that you can find everywhere. They're like very special habitat, like the alpine bogs, and they like to live with other penguin plants, like the round-leaved sandus and the pinguicula. vulgaris. Yeah. I guess if they're endangered, maybe the same level as just like other carnivorous plants. Yeah, but I think the wildfire is indeed influencing them. Yeah, because according to my experience, the box becoming smaller and smaller each time I went to the mountain. The forest is growing into the box, and they are becoming smaller and smaller right now. Yeah. Is there any other question? I think someone
Yeah,
have
John.
from the chat box. Yeah.
Yeah, John was wondering, are you suggesting that the Japanese species become occidentalists or its own?
Yeah. That should be what our conclusion. Yeah.
The carnivorous plant community is always excited to hear about a new genus. All right, we're getting lots of thank yous as you're well deserved. They appreciate it.
Yes, thank you very much. Yeah, if you have more questions, you can email me, and I'm glad to answer any questions.
Wonderful. All right, have a great day, everyone, and thank you, Dr. Qianshi Lin.
Yeah, bye.
Bye.
The International Carnivorous Plant Society wants you to be successful with your plants. We welcome growers just getting started all the way through professional scientists. We started an annual World Carnivorous Plant Day to celebrate these spectacular plants. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.